MES Marampally e-learning.
Hello everyone, my name is M. Murali Ramya, Assistant Professor, Department of Biosciences, MES College Marampally. Today, my topic of presentation is anaerobic culture methods. So, as we know that there are many microorganisms in the environment, but some organisms require oxygen, whereas some organisms do not require oxygen. And the presence of oxygen is detrimental to some of the organisms. Hence, depending on the requirement of oxygen, microorganisms are classified into several groups. And the anaerobic organisms are the organisms that do not require oxygen and they cannot survive in presence of oxygen. So, today we are going to discuss about various anaerobic culture methods. So, going to detail about the topic. Okay, let us move on to the session. So, we have discussed that there are various types of organisms that require oxygen for their growth, whereas some organisms do not require oxygen for their growth. So, based on the requirement for oxygen and toxicity of oxygen, these organisms were classified into certain groups. Among them, the first one is the obligate aerobes. These organisms grow only in the presence of oxygen, and the best example is Pseudomonas aeruginosa. And the next category is the facultative anaerobes. So, these organisms they can survive either in the presence of oxygen or in the absence of oxygen. In presence of oxygen, they respire, whereas in absence of oxygen, they do several fermentations. And the best example studied is the E. coli. And next is about the obligate anaerobes. So, these organisms they do not require oxygen for their growth, and at the same time even a trace amount of oxygen is toxic to these organisms. So, there are various groups under these obligate anaerobes, and some of the organisms are pathogenic forms also. So, the best examples are Clostridium, Ruminococcus, etc. So, next is about the microaerophiles. They grow best in the presence of oxygen lower than those of the air. That means the concentration of oxygen they require is less than that of the concentration of oxygen in air. And next organisms are aerotolerant anaerobes. So, they survive and grow in presence of small amounts of oxygen even though they do not require oxygen metabolically. They can tolerate the presence of small amounts of oxygen. So, bacteria have certain enzymes that convert oxygen within the cell into different chemical compounds, including the toxic substances like superoxide free radical, peroxides, which usually appear in the form of hydrogen peroxide and hydroxyl free radical. So, these compounds cause the oxidation of cell components and are lethal to the cell. And most aerobic, microaerophilic and facultatively anaerobic bacteria possess enzymes that convert these toxic substances to harmless compounds. Aerotolerant anaerobes have the enzyme superoxide dismutase, but they lack catalase to degrade hydrogen peroxide, whereas all obligate anaerobes they lack both the enzymes superoxide dismutase and catalase, and therefore they cannot convert the toxic forms of oxygen like hydrogen peroxide into non-toxic forms. Hence, a number of methods have been described for achieving anaerobiosis and for cultivating anaerobes. So, now we will see about the various methods of cultivation of anaerobic bacteria. Among them, the first is the evacuation and replacement of atmospheric oxygen in sealed jars. So, there are various jars available. So, the first one is the brewer jar. To cultivate anaerobes in the brewer jar, agar plates are inoculated with the organism and the plates are inverted and placed into the jar. So using scissors, cut off the corner of a foil packaged hydrogen generator and place this opened generator package into the jar in between the plates and the sides of the jar. Now add 10 ml of water into the hydrogen generator envelope and put the lid immediately and close the jar immediately. So by this process, the oxygen which is present inside the jar will be utilized by the hydrogen which is liberated in the gas pack, and this hydrogen combines with the oxygen to form water, and the reaction is catalyzed by the catalyst which is present on the lid of the chamber, which is nothing but the palladium. So with the help of palladium catalyst, the hydrogen reacts with the oxygen to form water, and the entire brewer jar is now placed in an incubator at the optimum temperature for the organism, with which the plates are inoculated for 24 to 48 hours. So next is about the gas pack system. So here a gas pack is available, and this the system consists of polycarbonate jar, and here the jar consists of a lid with a gasket to prevent the airflow, and it also contains two types of indicator strips. One strip is impregnated with reduction indicators such as methylene blue or risazurin, and whereas the other indicator is a biological indicator. So here disposable gas generating pouch, which is nothing but which consists of borohydride and sodium bicarbonate, is also incorporated in the jar, and it also consists of a palladium catalyst. So here inoculated plates or tubes are placed inside the polycarbonate jar or anaerobic container along with gas generator sachet and indicator strip, and is sealed completely. And in the presence of water, chemicals present inside the sachet, that is sodium bicarbonate and sodium borohydride, react chemically producing hydrogen and carbon dioxide gas. The hydrogen generated reacts with the oxygen present inside the jar and forms water, whereas the carbon dioxide liberated provides anaerobic environment inside the jar. And this reaction is catalyzed by the element palladium. The element palladium helps in the catalysis of oxygen and hydrogen to combine to form water. The palladium is present attached to the underside of the lid of the jar. So thus the jar will be under complete anaerobic environment. And the effectiveness of this gas packed anaerobic system can be checked by the following methods. One is the indicator system and the other one is the biological indicator system. So in the anaerobic indicator system, the methylene blue strip turns into blue color on exposure to oxygen, whereas in absence of oxygen or in anaerobic conditions, the methylene blue remains colorless. When we impregnate risazurin as an indicator, this risazurin appears pink in color in presence of oxygen, whereas in absence of oxygen it remains white in color. And the other biological indicator is the organism Pseudomonas. So we have already discussed that Pseudomonas aeruginosa is a obligate aerobic organism. So the growth of Pseudomonas inside the jar indicates that the jar consists of oxygen, whereas absence of growth of Pseudomonas inside the jar indicates that the jar is completely under anaerobic conditions. So this is the figure of the anaerobic jar which consists of the gas pack system. So you can see the piled up plates, Petri plates, and to the one side of the Petri plates you can see the indicator strip, and on the other side it is a gas envelope, gas pack. So here the lid consists of the palladium catalyst. And next method is about using chromium sulfuric acid method. So in this method, chromium metal powder is added to sulfuric acid and the reaction takes place. So when chromium metal powder reacts with the sulfuric acid, it forms chromium sulfate. At the same time, hydrogen gas is liberated. So the hydrogen gas thus liberated reacts with the oxygen which is already present in the jar and converts into water. So thus anaerobic conditions can be maintained. and at the same time the chromous sulfate in presence of sulfuric acid absorbs oxygen and is readily oxidized to chromic sulfate. Thus, chemical reactions involving only two reagents may serve two purposes. One is the displacement of air by the generated hydrogen, and other is the absorption of the residual oxygen by the chromous compound. Next, we are going to see about the various specialized techniques where evacuated jars are not used. So, among them first is the shake culture method. So, in this technique, sterilized and cooled medium is inoculated with a loop full of culture. Then the tube is shaken thoroughly and cooled rapidly and then incubated. So, after incubation, strict aerobes will grow on the surface of the tube, whereas anaerobes grow deeper inside the culture, whereas facultatively anaerobic organisms disperse throughout the medium and micro aerophiles grow near the surface of the medium. Next method is about the pyrogallic acid method. In pyrogallic acid NaOH method, pyrogallic acid and NaOH absorbs oxygen from the tube and creates an anaerobic atmosphere for the growth of the organism. So, in this method, at first the organism to be inoculated is streaked onto the surface of nutrient agar tubes, and next immediately after streaking the cotton plug is ignited, and after igniting it is pushed into the tube with the help of a glass rod. So, after the process, pyrogallic acid crystals were filled in the tube, and immediately after that 2 ml of 4% NaOH is added onto the top of the cotton plug, and then immediately after that it is replaced with a rubber stopper tightly, and these tubes were incubated in an inverted position at 37 degrees centigrade for 24 to 48 hours. So, this is the figure of the anaerobic culture technique, pyrogallic acid NaOH method. So, here you can see the inoculated slant upon which the culture is streaked upon, and upon the culture you can see the ignited cotton plug, and upon which a layer of pyrogallic acid crystals were seen, and upon which we have added 2 ml of 4% NaOH, and then the tube is immediately tightly sealed with a rubber stopper, and these tubes are incubated in an inverted position. Next is about the paraffin plug technique. So, in this technique, sterilize the liquid paraffin at 180 degrees centigrade in an oven for 1 hour. So, after sterilization, pour the paraffin over the slope of the slant in such a way that the paraffin forms a layer of about ½ inch above the agar surface, and then these tubes were stored in an upright position at room temperature. So, next we will see about the cultivation of strict anaerobes or obligate anaerobes. So, these obligate anaerobes cannot survive in presence of small amounts of oxygen. So, there are various specialized techniques for cultivating these strict anaerobes. So, among them the first is the roll tube method. So, this was developed in which agar medium was distributed as a thin layer over the internal surface of test tubes charged with an anaerobic atmosphere for the isolation of obligate anaerobes. In this method, exposure of bacteria and culture media to air is avoided by displacing the air in the culture vessel with an oxygen free gas such as carbon dioxide, hydrogen, nitrogen, or a mixture of these gases. So, in this method, inoculation is done with a transfer loop held against the agar surface as the tube is being rotated by a motor. By starting at the bottom and drawing the loop gradually upward, the inoculum becomes thinned due to the point where well isolated colonies can be developed, or where these isolated colonies can be seen, and vessels are stopped under conditions preventing access of air. The cultures require no special incubators and can be removed and examined with no anaerobic precautions if tightly kept. So, this is the picture of the roll tube method. So, here you can see the tube which is kept on a shaker or a motor. So, when the motor is supplied with the electric current, it rotates, and along with the rotation of the motor, the tube also gets rotated. So when we keep the inoculating loop at the bottom of the tube, and when we draw the loop upwards, we can streak onto the surface of the medium, and along with that you can see a carbon dioxide generator at the side, so that we can get complete anaerobic conditions in the roll tube method. So next is about the anaerobic chamber, also known as glow box. So the anaerobic chambers provide a strict anaerobic atmosphere of 0 to 5 parts per million using a palladium catalyst and hydrogen gas mix of 5%. So here culture media are placed within the chamber by means of an airlock, which can be evacuated and refilled with hydrogen. After being rendered oxygen free, the media are inoculated within the chamber by means of glow pods and incubated within the chamber. So next, a special cuff and sleeve vacuum system allows the operator to work bare-handed. So here the glow pods means it also consists of surgical gloves. The arm port plugs are usually kept idle when they are not in use. So this is the figure of glow box. So you can see the glow pods, so through which a person can put his hands into the glow pods, and you can inoculate the plates, and you can see an anaerobic chamber nearby the glow pods. So after inoculating the plates, it can be immediately sent to the anaerobic chamber, where it can be incubated under anaerobic conditions. Next is about the candle jar. It is mainly used to cultivate organisms like aerotolerant anaerobes and micro aerophiles. And this is a glass jar containing a sealed jar. It is a sealed jar, and here a candle is lighted, and the jar is it is kept inside the jar, and the lid is closed immediately. And the lighted candle in the jar utilizes the initial oxygen, and when the candle goes out, a 3 to 4% carbon dioxide atmosphere is established within the jar. And here also you can use indicator strips like methylene blue. So when the jar consists of oxygen, the methylene blue strip turns blue in color, whereas under anaerobic conditions it remains white in color. So this is a figure of the candle jar. So you can see the petal plates inside the jar, upon which we have kept a lighted candle, and immediately after keeping the candle, the lid should be closed. So when you close the lid, you can see that the air, the oxygen present inside the jar is utilized by the lighted candle, and when the candle goes out, it liberates some carbon dioxide, which provides anaerobic conditions for the growth of organisms present inside the jar. So it also consists of an indicator system. So with this we have come to the end of the session. I hope you understand well about various anaerobic culture techniques. Thank you.